or what is the respiratory substrate? Any idea? Martin, what are the respiratory substrate?
Yes, sir.
Sugar. Okay, fine. That is also a respiratory substrate. So look, first try to understand what is written over here. You will have to jot down first, but before you join Martin, I told that respiration is similar in both animal as well as plant, right? Like there is no difference in the. There are some differences, no doubt, but there is not major difference. So uh, there are some minor differences between the plant and animal respiration, but normally the process is similar. Look, first I'm trying to make you understand what respiration exactly is, or what respiratory substrate is. Look, uh, respiration is the process in which burning or oxidation of any substrate or substance takes place. So some of the substance they burn inside mitochondria. Mitochondria is a generator, right? It works like generator. So how does generator work at your home? You fill a tank with the some fuel, right? That fuel start burning, and due to burning of that fuel, what happened? Heat is generated, energy is generated, electricity is generated, right? Different vibration energy, different kind of energy is generated. So from where these energies are coming? These all energy are coming due to burning of the fuel, right? If it is petrol generator, you will burn the petrol, so substrate is petrol. If it is diesel generator, the substrate is diesel. If it is ethanol generator, the substrate is ethanol. So what is the substrate? Substrate is fuel, right? Same way in our body, in our mitochondria, the substrate is that compound or that chemical which will burn or which will oxidize. Due to burning of that chemical, energy generation and carbon dioxide will be coming out, right? So that is called respiratory substrate. So any kind of the chemical which burn out in our body and provide us energy, we call that respiratory substrate. So try to understand over here. I have written over here, respiratory substrate, it burn during the process of respiration and basically burn in mitochondria. Carbon dioxide come out and energy is generated. This part is called respiration. Now, I have told you that respiratory substrate are those substances which burn out. What can be respiratory substrate for human, for plant, or for all living being, all eukaryote? Normally, let's talk about the human or let's talk about the plant. The best respiratory substrate is carbohydrate. When I say carbohydrate, that is sugar. Martin told us that sugar is respiratory. Yeah, sugar is a type of respiratory substrate. But it is not only sugar which is respiratory substrate. Anything which is going to burn inside the mitochondria, it is providing us energy and the carbon dioxide is coming out. That is called respiratory substrate. Look, carbohydrate, simply we call it sugar. So this is a respiratory substrate. So what will happen? First, carbohydrate is a complex. We have already studied the biomolecule part. So there are you know that what is a polymer, what is a monomer, what is fat, what is protein. So I don't think that I need to mention over here. Carbohydrate will break down in our body in the form of glucose. Then what will happen? Glucose will break down into pyruvate, and then pyruvate will break down into acetyl coenzyme. Right? Why it happens? Why glucose break down to pyruvate? Why pyruvate break down to acetyl coenzyme? I will tell you. So finally, this acetyl coenzyme A, it burn and provide us energy. Right? The same way, fat. Right? Fat break down into fatty acid, and fatty acid break down to acetyl coenzyme A. And this acetyl coenzyme A burn inside our mitochondria in the Krebs cycle and provide us energy. Look. Then what is the third thing? Protein. Normally, protein is not respiratory substrate, but sometimes it works as a respiratory substrate. So protein will break down into amino acid. When protein break down as a respiratory substrate or protein is used as a respiratory substrate, right? We call it cytoplasmic respiration, right? It break down into amino acid and amino acid break down into organic acid. And organic acid produces energy, right? Then the fourth respiratory substrate is organic acid. Organic acid directly used as a respiratory substrate. They need not to break down some monomer, right? So what are the basically respiratory, overall respiratory substrate? What can be respiratory substrate? So carbohydrate, fat, protein, or organic acid can be respiratory substrate. They break down, and due to breaking down and due to their oxidation, we get the energy. Look, very important. Burning of carbohydrate and fat is called floating respiration. Normally, the burning of carbohydrate takes place when carbohydrate is completely consumed. Preferably, body require carbohydrate for the burning. But when carbohydrate get consumed, then it comes to the fat, and fat is utilized. You know, the burning of fat takes place when we have less carbohydrate in our body. So burning of, when respiratory substrate is burned in the form of carbohydrate or fat, we call it floating respiration. But when burning of protein and organic acid takes place, we call it, when burning of protein takes place, we call it cytoplasmic respiration. Why? Because when protein burning start, on that time body start eating itself, right? How it takes place, right? Suppose that there's a normal human being. He or she is in, uh, uh, you can say, uh, in starvation situation, or you can say in fasting, right? So what will happen? First, the complete carbohydrate of body will break down, and the person is not taking even a drop of the food. After the consumption of carbohydrate of whole body, what will happen? Now it comes to the fat. How long a person will survive, it will depend on the amount of the fat is present in the body. It means the skinny kind of the people who are thin, or who have thin body, they won't survive for longer time, but a fat man will survive for the longer time, because he or she have the fat for burning for the longer time. After in 1 month, 2 months, when complete fat is consumed in the form of energy, then it comes to the protein. Now body will start breaking down protein, and when protein start when burning of protein start, a person will die, right? So that's why we call it, then the body will start eating itself, cell will start eating itself, right? Mitochondria will start breaking its own component, right? That's why we call it cytoplasmic respiration, right? So this is all about the respiratory substrate. Tell me, is it clear or not? What is the respiratory?
Yes, sir.
Martin, that's clear to you? Hello, Martin.
Yes, sir.
Okay, so first what you have to do, you have to jot down all these things in the same way. No, it's a bit clumsy, but you have to write it down in clear way, right? Write down right now. Then I will dictate. Put the heading respiratory subject. Let me know when you note it down. Done?
Yes, sir.
Okay. Martin, done?
Yes, sir.
Okay. One more thing I want to explain you about the ATP. By the way, I have explained it in the part of biomolecule as well, right? So Alia is there. Okay, fine. So try to understand. Look, ATP is called energy currency. Why? Energy is stored in our body in the form of ATP, right? So what is the adenosine triphosphate, right? So this is adenosine, right? This is adenosine. So that is a nitrogenous base, we know. Adenosine, this is structure of it. There are three phosphate, one, two, three. There, that's why we call it adenosine triphosphate, because there are three phosphate. These bond, I have shown the bond like this way, right? Why? Because when the bonds are breakable bond or they can be broken easily, so we use like this kind of the symbol, right? Wavy kind of symbol. So this is the adenosine triphosphate. What happened? This is an energy molecule, energy currency. When we require energy, this last bond breakdown, last phosphate gets separated. So what happened? Last bond broken down. Phosphate, Pi means inorganic phosphate. This Pi represent inorganic phosphate. We call it inorganic phosphate. So this is an organic phosphate. It gets separated, energy is liberated, and we use that energy in working. Now there are only two phosphates, so we call it ADP, adenosine diphosphate. In exceptional cases, not regular. So normally ATP break down into ADP and energy is liberated, right? Then what happened in the emergency situation? One more bond breakdown and phosphate come out, energy is liberated. Now only one phosphate is left. We call it AMP, adenosine monophosphate, right? The reverse process takes place when energy is stored. So that's how the energy is spent. What will happen? This AMP will make a bond with one more phosphate, add one more phosphate. We call it adenosine diphosphate, right? In ADP will become ATP. How? One more phosphate will join, and ADP will become ATP. So that's how the energy generation reverse process is the energy generation process, and this process is called what? Energy extension process, expenditure process. Got it? Clear?
Yes, sir.
Okay. So please note down this diagram as well. Alia, clear?
Yes, sir.
Please note down this. What is the ATP, ADP, and AMP? Done.
Done, sir.
Okay. So please start writing now, right? What are the respiratory substrate? Because if you'll get the question, what are the respiratory substrate, then you have to first define the respiratory. So what is the definition? Respiratory substrates are those organic substances which oxidized during respiration, oxidized during respiration to liberate energy inside living cells. Full stop. The common respiratory substrate substrates are carbohydrate. The common respiratory substrates are carbohydrate, protein, fat, and organic acids. Full stop. Next point. The most common respiratory substrate is glucose. Next point. Next paragraph, in fact, protein are used in respiration rarely as during germination of seed and spore. These are the rare situations. Next point, it is very important. Respiration involving protein as respiratory substrate is called protoplasmic respiration, while when carbohydrate and fat are used as respiratory substrate, while carbohydrate or fat are used as respiratory substrate, we call it floating respiration. Right? So this was all about the respiratory substrate. What are the respiratory substrate? The same thing is given in your first part, like first and second page of the this one NCRT, right? Now come to the how the respiration takes place. What are the type of respiration, right? Here directly given to the glycolysis in your NCRT, they have directly jumped into the glycolysis, right? And that is not the good sequence, glycolysis. And in the last, they have mentioned what is the anaerobic respiration and fermentation. So let me make you clear, and you have to study like this way in sequence. The second thing is the type of respiration. So you have to put the heading type of respiration. But first, let me make you understand, right? And you have to copy it as it is, right? Look, I will explain what is the aerobic and anaerobic. Don't jump directly to writing now. So first, aerobic respiration and anaerobic. From the name itself, it is clear in aerobic respiration, the burning takes place in presence of oxygen. And in anaerobic respiration, the burning takes place in absence of oxygen. Yes, burning also takes place in absence of oxygen. There are some organisms which can generate energy without utilization of oxygen, right? Look. Aerobic respiration normally takes place in plant and animal in general, right? The burning of food take place in presence of oxygen, right? What happened? So normally glucose, we have already written that normally the respiratory substrate is glucose. So everywhere, wherever we will study the respiration, we will always take the glucose, right? Or carbohydrate as a respiratory substrate. So glucose is a respiratory substrate. The burning of glucose takes place in presence of oxygen. So C6H12O6 plus oxygen, formation of 6 molecule of carbon dioxide will take place, 6 water molecule will be formed, and 686 kilocalorie energy is generated. Now, what is the anaerobic respiration? Burning of carbohydrate or burning of food or substrate takes place in absence of oxygen, right? It can be of two types, right? So it takes place, normally it takes place in bacteria, fungi. It also takes place in human muscles when scarcity or supply of oxygen is less in our body, right? The muscles or the body part which do not get optimum amount of the oxygen or scarcity of oxygen is there, that particular part or that particular cells of the body, they start anaerobic respiration, right? So when this anaerobic respiration takes place in bacteria and fungi, so what actually happened? So normally, what actually happened? This bacteria or in fungi, respiration takes place, and this glucose breakdown in presence of enzyme, glucose breakdown into carbon dioxide and ethyl alcohol in bacteria and fungi, right? This is also called fermentation, right? So wherever the fermentation takes place, normally bacteria and fungi are used for fermentation. When you make the bread, when you make the wine, when you make the beer, when you make the whiskey, right? All that kind of the fermentation, or in fact, when you make the idli or dough for something. So on that case, the bacteria and fungi are used, right? These bacteria and fungi, they have particular enzyme which break down glucose into what? Carbon dioxide. So two molecules of CO2 are created, and in place of water, try to understand, in aerobic respiration, water formation taken place. But in anaerobic respiration, formation of alcohol taken place, right? And the less amount of energy is generated, 59 kilocalorie energy. That's why I have written that this is the fermentation. So in case of fermentation, this kind of reaction takes place. But in human body, when anaerobic respiration takes place, particular cell or particular muscle, when during the hard work, during the running, during heavy exercise, and that time, what happened in human body, ethyl alcohol formation do not take place. That is dangerous for our body. So what happened? Again, the carbohydrate or glucose burned, and due to burning of carbohydrate or glucose in presence of particular enzyme, formation of lactic acid takes place, C3H6O3, right? So lactic, in place of ethyl alcohol in our body, formation of lactic acid takes place, and 36 kilocalorie energy is generated, meaning very less amount of energy is generated. And along with that, what happened? This lactic acid deposit in our muscles where anaerobic respiration is taking place, and we feel muscular cramp because of this lactic acid. When we take rest, after taking rest, what happens? This lactic acid breaks down, and due to breakdown of the lactic acid, we feel relaxed and muscular cramp is removed. So this is the type of, all about the type of respiration. Tell me, is it clear or not?
Yes, sir. So can you explain the bacterial part
Yeah.
again?
In bacterial, what happened? In bacteria and fungi, when there is, suppose that you have put a bacteria, particular dough, or you have put yeast or fungi particular dough, what will happen? It will release enzyme outside. It will break down glucose into 2 carbon dioxide molecule, and ethyl alcohol formation will take place. Ethyl alcohol will evaporate, right? And then formation of 59 kilocalorie energy takes place. So this bacterial anaerobic respiration, this fungal anaerobic respiration is also called fermentation.
Okay, sir.
Clear?
Yes, sir. Yes. Thank you,
So,
sir.
when you make the bread, right, you take a dough, you mix yeast, then fermentation takes place. Those large size of the hole which are inside the bread, they are due to the removal or escape of the carbon dioxide. So carbon dioxide try to escape, and that make those holes, right?
Yes, sir. Yes.
Okay, fine. So please note it down, both type of the respiration over here. Copy and remember it.
Done, sir.
Done? Okay, Martin, is it done?
Yeah.
Okay, great. So now come to the point that how does respiration takes place. So there is nothing to dictate over here. So RQ is the last part of your respiratory question. I'll tell you last. This diagram is very important. I want you to learn it by heart, and I want you to understand it by heart. Right? Try to understand. I want your 100% focus, because you will not get this diagram anywhere. Right? Uh, this diagram is complete information about the respiration, what respiration exactly is. And this is the complete summary of basically complete chapter. Right? Try to understand what is this. How does it take place? Look, whatever you eat, sugar in the form of sugar, like we eat chapati, we eat bread, whatever we eat grains, we eat pulses. So those all different form of sugar, like carbohydrate. When we eat, they goes into stomach, then in intestine, and they break down. So finally, when digestion takes place, they break down in the form of glucose, right? So glucose is a respiratory substance. That glucose is absorbed by our intestine, right? When intestine absorbs it, that is directly mixed into our blood, right? Blood circulation takes place. Blood distribute this glucose every nook and corner of our body, right? And that glucose goes to cell, right? This glucose enter inside our cell, where in cytoplasm or protoplasm. Now the story starts, right? But burning of glucose have to take place in mitochondria. But glucose is a larger molecule. It is a 6-carbon compound. You can see over here. It is 6-carbon compound. So our plasma membrane give entry to glucose. Such a larger molecule can enter inside the plasma membrane. It can reach into the cytoplasm, but it cannot enter into mitochondria. We know that mitochondria is made up of two membranes, outer membrane and inner membrane. It cannot go. Why? Because the size of the glucose is larger, right? It cannot go through, right? Then what is the mechanism? So before it enter into mitochondria, mitochondria can allow a molecule which is made up of 3 carbon, not the 6 carbon. So the glucose is broken down into pyruvate. Try to understand. Glucose is a 6-carbon compound is broken down into pyruvate, which is 3 carbon. This can enter into mitochondria. So the process of breaking glucose into pyruvate molecule, glucose is a 6-carbon compound. It glucose breaks down into 2 pyruvate molecule, and pyruvate is 3-carbon compound, right? This process is called glucose, and this complete process goes on in cytoplasm. Is that clear?
Yes, sir.
Okay. So what is the glycolysis? Where does it goes on? It goes on into protoplasm or cytoplasm, basically in cytoplasm. Why? Because glucose cannot enter into mitochondria, or you can say directly that mitochondrial membrane are not permeable for glucose, right? Then what will happen? It will break down into 3-carbon. When glycolysis goes on, or this breaking process goes on, what will happen? Formation of ATP will take place, and net 8 ATP will be produced in glycolysis. So there are two benefits. First, it will break down larger molecule into smaller molecule, means glucose into 2 pyruvate, and along with that also energy gain is taking place, and 8 ATP are net produced. Now what will happen? These both of the pyruvate will enter. Now mitochondria membrane is permeable, or it allow the pyruvate molecule to enter. Now the pyruvate will enter inside the mitochondria. Inside the mitochondria, after crossing outer membrane and inner membrane, there is a gel-like substance. We call that matrix. Yes. In matrix, this Krebs cycle goes on. Krebs cycle is the process when what will happen during this process or during this cycle, complete burning of glucose will take place and energy generation will take place. But pyruvate is a 3-carbon compound. It can enter inside the mitochondrial membrane, but it cannot participate in Krebs cycle. So it have to convert into a 2-carbon compound, that is acetyl coenzyme. So inside the matrix, pyruvate will break down into 2-carbon compound acetyl coenzyme. This acetyl coenzyme A will break down into this Krebs cycle, and due to breakdown of acetyl coenzyme A, the carbon dioxide and energy will be generated. Energy will be generated in the form of ATP, NADPH, and FAD, right? This energy which is generated in Krebs cycle is equivalent to 30 ATP, right? So 30 ATP generated in Krebs cycle, right? And 8 ATP generated in where? It is generated in glycolysis. Total 38 ATP generated due to burning of one glucose molecule. That's why we say due to burning of one glucose molecule, 38 ATP are generated. This is the complete balance sheet of energy balance sheet of complete respiration, right? GDP is equal to ATP, right? 1 NADH is equal to 3 ATP. I have told you multiple times. 1 FADH is equal to 2 ATP. That is also we know, right? And one glucose molecule generate 38 ATP energy in the process of aerobic respiration. Is it clear or not?
Yes, sir. Yes.
If it is
Yes, sir.
crystal clear to all of you, Alia, is it clear? Martin,
Yes,
what about?
sir.
Okay, great. So I request you to note down as it is, right? Right now. Please do it.
Sir,
Yes.
sir, can I leave at 5 o'clock?
Yeah. Do you have some emergency?
Sir, there's the parent teaching meeting in school.
Okay, fine. No issue. But take the backup of the class, right? Because this is
Okay,
very
sir.
important, right?
Okay, sir. Yes, sir. Yes, sir. Sir, I noted this down.
Yeah, sure. You can leave.
Done, sir.
Done? Okay. Martin, done?
Yes, sir.
Okay. So now the next is that is the uh, glycolysis process, right? We need to understand what is the glycolysis. Glycolysis is also known as the EMP pathway because it was discovered by three scientists, Emden, Meyerhoff, and Paranj, right? J. Paranj, right? So Emden, Meyerhoff, and J. Paranj, they discovered it. That's why we call it EMP pathway, right? So glycolysis can be also defined as EMP pathway. Now, where does glycolysis take place? Uh, After my explanation, you have to note down only those things which have been underlined with the red color, right? And whatever is written along with that, that is more important. Try to understand first, because here understanding is more important in this chapter. Cramming is not important, right? Look, first step in first reaction, glucose breaks down into glucose 6-phosphate. Due to breakdown of glucose, consumption of energy takes place. So we divide glycolysis in two parts, where first preparatory phase, try to understand, I want your 100% focus over here. Look, we divide glycolysis in two parts, preparatory phase and payoff phase. In preparatory phase, glycolysis take energy, consume energy, and in payoff phase, it start giving us back energy, right? That's why we call it payoff phase and preparatory phase, right? So first phase, from here to here, I have broken down into two part here, from here to this point, right? From here here to this, right? So till this point, we call it what? Preparatory phase, and below this point, from glyceraldehyde 3-phosphate G3P onward. This is called payoff phase. Look, glucose breaks down into glucose 6-phosphate. When glucose breaks down into glucose 6-phosphate, 1 ATP is consumed. I have written over here minus, right? If you do not remember the enzyme, it's okay. That's why I have not underlined the enzyme. I know you will forget it. Glucose will break down to glucose 6-phosphate. Now the glucose 6-phosphate will convert into fructose 6-phosphate. Both are isomer of each other. That's why the enzyme which is utilized over here is isomerase. In the presence of this isomerase enzyme, glucose 6-phosphate will convert into fructose 6-phosphate. Right? Again, now the fructose 6-phosphate break down into fructose 1,6 biphosphate. Again, 1 ATP is consumed over here. Minus 1. Again, I have written over here minus 1. Right? Again, one more ATP is consumed over here. Now, fructose 6-phosphate, 1,6 biphosphate. Here onward, the payoff phase will start. Fructose 1,6 biphosphate break down in the presence of aldose enzyme break down into two compounds. Right? What are they? DHAP. In short, we call it DHAP. What is DHAP? Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. They are interchangeable or interconvertible. Right? Again, in the presence of isomerase enzyme. Right? They are isomer of each other. Right? So dihydroxyacetone phosphate do not participate directly into glycolysis. Hence, it again convert into glyceraldehyde 3-phosphate. In short, we call it G3P. Right? Now, glyceraldehyde 3-phosphate will break down into 1,3-biphosphoglycerate. Right? When glyceraldehyde 3-phosphate break down into 1,3-biphosphoglycerate, again 2 NADH will be produced. Right? And I have already told you that NADH is equal to what is 3 ATP. 1 NADH is equal to 3 ATP. So I have written over here 2 NADH molecule are produced. Again, 1,3-biphosphoglycerate. So now energy generation started. That's why we call it payoff phase. 1,3-biphosphoglycerate breaks down. Try to understand. 1,3-biphosphoglycerate break down into 3-phosphoglycerate. Again, 2 ATP are generated. Energy generation takes place. 3-phosphoglycerate change into 2-phosphoglycerate. No energy is generated. 2-phosphoglycerate change into phosphoenol pyruvate (PEP). Right? Again, uh, and, yes, no energy generation takes place. Right? Phosphoenol pyruvate (PEP) change into 2-pyruvate, and again NADH generation takes place. Means NADH formation takes place. So this complete process is called glycolysis, and from one molecule of glucose, formation of a 2-pyruvate molecule takes place. This is called glycolysis or EMP pathway. So please copy this one as it is. Right? Try to understand. Look, how many energy are consumed? Look, -1. So I have written -2 ATP are consumed. This one -2. Try to understand. -2 ATP are consumed. Now, how many NADH are produced? 2 NADH are produced. Right? Okay. How many? Again, plus 2 ATP. I have written over here 2 ATP. Again, plus 2 ATP here. Right? NADH is equal. Sorry. Where? Yes. Here the two. Again, here the 2 ATP are produced. Again, plus 2 ATP are produced. So 2 NADH is equal to 6 ATP. Again, look, -2 ATP. 2 NADH is equal to 6 ATP. 6 ATP plus 2 ATP. 2 ATP plus 2 ATP. So 6, 2 plus 2 total 10 ATP minus 2 ATP. Net gain is 8 ATP. That's how 8 came over here, right? So that is energy balance. Tell me first, is it clear or not? If not clear, ask me right now. Is that clear? Martin, Alia, both of you?
Yes, sir. Yes, sir.
Okay. So please note down it right now as it is. And you have to note down only those part where I have underlined, and the same way you have to note down the energy balance sheet separately, right? This is the energy balance sheet. The side, please note down the energy balance sheet. Let me know, Martin and Alia, when you note it down.
Okay.
Done.
Yeah.
Yeah, please note it down carefully because this is important question. If they will ask you glycolysis, this will be a full mark question and a bit difficult too.
Done, sir.
Done. Okay. Martin, is it done?
Yes.
Okay, fine. So start noting down. So everything understood?
Yes, sir.
Okay, great. So start noting down. Put the heading glycolysis. It is also called EMP pathway. It is also called EMP pathway because it was discovered by three German scientists because it was discovered by three German scientists, Gustav Amden. I have written the spelling over here. Gustav Amden, comma, Otto Meyerhoff, and J. Parans, in 1930. Full stop. Next point. Glycolysis is the process of partial oxidation of glucose into two molecules of pyruvic acid through a series of 10 enzyme mediated reactions, releasing some energy in the form of ATP and NADH2. ATP and NADH2. Full stop. Next point. It occurs in cytoplasm. Next point. Glycolysis has two phases. Preparatory and payoff phases. Full stop. In preparatory phase, glucose is broken down into glyceraldehyde 3-phosphate. In preparatory phase, glucose is broken down into glyceraldehyde 3-phosphate. Full stop. In payoff phase, G3P or glyceraldehyde 3-phosphate, in payoff phase, glyceraldehyde 3-phosphate changes into pyruvic acid and energy is produced. Full stop. Next point. In glycolysis, net gain of 8 ATP molecule takes place. That's it. So this was all about the glycolysis. You have to note it down, like when you get the question in the form of describe the glycolysis with the help of flow chart or with the help of a skeletal diagram. Then you have to write down this language, and after that you have to write the this glycolysis skeletal diagram, right? And in exam, if they ask you to write it down this energy balance sheet, then you have to note down. Otherwise, this is for your understanding, but they can ask you, right? And this will help you out even after your this exam as well, right? But this complete diagram, and you have to like describe which one is the preparatory phase, which one is the payoff phase, and that's how you have to write it down. Now come to the Krebs cycle. What will happen? So where the Krebs cycle take place, that I have already explained, right? And how does it take place? Where does it take place? Inside the mitochondria matrix. So in glucose, when glucose breaks down into pyruvate, 8 ATP energy is produced, and when this pyruvate changes into oxaloacetic acid, finally, then 30 ATP is energy is generated, right? So this is the Krebs cycle or TCA cycle. We call it tricarboxylic acid cycle, right? Because the first compound is tricarboxylic compound, right? So that's how you can call it TCA cycle or Krebs cycle. Look, what happens and how does it take? Here, one glucose molecule breaks down into 2 pyruvate. Pyruvate in glycolysis. After that, this pyruvate decarboxylation of pyruvate takes place. Mean one carbon dioxide is removed, one carbon molecule, carbon dioxide removes. This is called decarboxylation. Why? Because carbon dioxide is getting removed, right? So when one carbon will come out of pyruvate is 3 carbon molecule, and from here the 1 carbon is get out or released. Then what will happen? It will become a 2 carbon compound. So 2 pyruvate will convert into 2 molecule of acetyl coenzyme A, right? Now this acetyl coenzyme A will start working in this cycle, right? Again, NAD will produce NADH2. So here the formation of 3 molecule of NADH2 takes place, right? So 3 molecule means equal to no, sorry, not 3 molecule. This is the formation of NADH2 will take place, right? So here the formation of NADH2 takes place. Now what will happen? Acetyl coenzyme A will break down into citrate. Citrate is, it will become citrate. Citrate is a 6 carbon compound. Then from citrate, formation of isocitrate will takes place. Again 6 carbon compound. Then when isocitrate will change into alpha ketoglutarate, again decarboxylation will take place and formation of NADH will take place. I mean, this is the second place when energy generation will take place, right? Again, the alpha ketoglutarate is a 5 carbon compound. This will changes into, it will break down into succinyl coenzyme A. Again, decarboxylation will take place, means 1 carbon dioxide will go out, and this is a 4 carbon compound. Again, formation of NADH will take place. So look, here the production of energy taken place. Again, here the energy production taken place. Again, here the NADH formation will take place. Again, acetyl coenzyme A, succinyl coenzyme A, which is 4 carbon compound, is breakdown into succinate. In presence of succinyl coenzyme A synthase enzyme, right? Formation of GDP will take place. So GDP will take breakdown changes into GTP. So GTP is equal to ATP, that I have already explained to you. So when the succinyl coenzyme A will change into succinate, again the energy generation will take place. Again, this succinate will change into fumarate. Succinate is 4-carbon compound and this is 4-carbon compound. In the presence of enzyme succinate dehydrogenase. So what will happen? From FAD to FADS2 formation will take place. 1 FADS2 is equal to 2 ATP. So here I have told 1 GTP is equal to 1 ATP. 1 FADH is equal to 2 ATP and 1 NADH is equal to 3 ATP, right? Again, formation of fumarate take place. So from when fumarate formation take place, again FADH2 formation will take place. When fumarate will change into malate, again a 4-carbon compound, no energy generation take place. When malate will change again into oxaloacetate, right? This is oxaloacetate, which is 4-carbon compound, right? Then again NADH formation take place, means energy generation will take place. Look, now this oxaloacetate, when again the cycle restart. This 4-carbon compound oxaloacetate, it combined with acetyl coenzyme, right? From where? Which is coming from the pyruvate. When this 4-carbon compound will combine with 2-carbon compound, then formation of again citrate will take place, right? How does citrate formation take place? This acetyl coenzyme have combined with oxaloacetate, right? When they combine, 2-carbon compound combined with 4-carbon compound, formation of citrate takes place. This cycle is called, tricarboxylic acid cycle or citric acid cycle, right? Because citrate or citric acid is the first stable compound, and this what is discovered by Hans Krebs. That's why called Krebs cycle, right? So this complete process is the burning of acetyl coenzyme or burning of glucose, and that's how the generation of energy takes place. Okay. If you have doubt anywhere. Martin, is that clear? Martin, note it down.
Yes, sir.
Complete, note it down. Okay, fine. So write it down. What is the TCA cycle? Put the heading Krebs cycle or TCA cycle. Krebs cycle was discovered by Hans Krebs. Krebs cycle was discovered by Hans Krebs. H-A-N-S, Hans Krebs. Krebs cycle was discovered by Hans Krebs in 1937. 1937. He got the Nobel Prize for same. He got the Nobel Prize for same. Full stop. It occurs inside the matrix of mitochondria. Full stop. The cycle is also named citric acid cycle or citric acid cycle or TCA, tricarboxylic acid cycle. After the initial product. Next point. Krebs cycle is stepwise. Krebs cycle is stepwise oxidative and cyclic degradation of acetate which is derived from pyruvate. One more thing, write down this reaction as well. Glucose plus 4 ADP plus 4 H3PO4 plus 4 NAD plus 2 FAD. Then glucose will break down completely into carbon dioxide, 6 CO2, 4 ATP, 10 NADPH, 10 hydrogen plus means proton, and 2 FADH2. This is the complete reaction of glycolysis, the Krebs cycle. Note down, Martin. It's complete reaction. Done, Martin?
Yes.
Note down after the second note down. A molecule of glucose, glucose yield 2 molecule of 2 molecule of NADH2, comma 2 ATP and 2 pyruvate during glycolysis. Full stop. The 2 pyruvate completely degraded in Krebs cycle, form 2 ATP, comma 8 NADH2 and 2 FADH2. Full stop. That's how the complete oxidation of a glucose molecule generate 38 ATP energy. That's it. So this was all about the this is all about for today.